This is the MEDS platform, a targeted nanoparticle-based system for precision drug delivery to the kidneys and other organs. Polycystic kidney disease is a genetic disorder that causes many cysts to grow in the kidneys and disrupt kidney function. If not successfully treated, PKD leads to kidney failure and a significantly degraded quality of life that requires dialysis and kidney transplantation. Many potentially useful drugs are limited by their inability to get past various physiological and biological barriers to achieve maximum therapeutic efficacy at the disease site while limiting off-target side effects. The Elferman Institute, in collaboration with Professor Eunji Chung at USC, is addressing this unmet need by translating her micelle solution out of the laboratory and demonstrating it as a therapy for polycystic kidney disease, or PKD. The platform is based on the unique properties of small molecule drugs carried by nanoparticles called micelles. One major advantage differentiating this micelle platform is its small size, each around 10 to 15 nanometers in diameter. This allows the micelles to passively traverse tissue membrane barriers to enable unprecedented drug accumulation in the kidneys. A small molecule drug candidate is either attached to the surface or loaded into the core of the micelle for targeted delivery to the kidney. Oral drug delivery is an essential convenience for the treatment of chronic diseases such as PKD. To be effective, an orally administered drug must not be metabolized by the stomach and intestines and must traverse the intestinal mucosa and into the bloodstream. Once in the bloodstream, the drug must circulate until it finds its target tissue and then be selectively and sufficiently absorbed at the targeted tissue. Professor Chung has developed a unique chitin shell that encapsulates the micelles and that is able to resist stomach and intestinal metabolism, crosses the mucosal barrier to release micelle nanoparticles that reaches the bloodstream and can circulate until targeting the kidneys. PKD affects about 12.5 million people worldwide and 700,000 people in the US, while all chronic kidney diseases affect 700 million people worldwide. Currently, there is only one FDA-approved drug for PKD, and many of the PKD drugs under investigation also have side effects due to the lack of high-affinity organ targeting. As seen from small animal studies data, meds have a remarkable ability to precisely target the kidney and can bind specific kidney cell receptors. This encouraging result demonstrates our micelle's ability to target the kidney for treatment of polycystic kidney disease. Recognizing the significant potential for an orally administered and kidney-targeted drug delivery system, we are currently exploring other candidate drugs. Our goal is to identify drug candidates that can benefit from the increased targeting efficiency, resulting in reduced side effects and reduced drug load in off-target tissues, or additional use indications when delivered with meds. Drugs under advanced clinical evaluation include rapamycin, bardoxolone methyl, and metformin, which have been shown to reduce the size of kidney cysts in PKD. Other kidney diseases and complications that could benefit from this targeted delivery platform include kidney cancer, Alport syndrome, and glomerular nephritis. This convenient delivery system would enable the substitution of other drugs for the ones being developed for kidney diseases that must currently be given by injection. Moving these injectable drugs into oral meds formulations are expected to significantly improve both clinical compliance and clinical outcomes for patients.